Okay, think about this. You grab milk from the fridge, maybe open a can of beans for dinner, or, you might be heading for a medical procedure with sterilized tools.
Mm-hmm.
We just sort of trust that this stuff is safe,
Right.
that there aren't dangerous microbes lurking around. But how does science actually make sure of that?
Yeah, it's this invisible shield we rely on.
Exactly. Like, how do we know that just using heat is enough to wipe out these tiny threats reliably every single time?
It's a great question, and it's something we often take for granted. But behind that trust, there's some really precise science, a deep dive, really, into how heat actually impacts microorganisms.
And how to measure it, predict it.
And apply it, yeah, apply that knowledge effectively on a huge scale.
Right. And that's exactly what we're digging into today. We've got some great source material here that really lays out the fundamental principles of microbial death by heat, the kinetics of it almost.
Think of it like unlocking the rulebook for thermal processing safety.
So our mission today is to unpack those key ideas, the concepts, and also the actual calculations researchers and industry use. Because it's definitely not just about cranking up the heat.
No, not at all. It's about figuring out exactly how much heat and for how long, and why nailing that down needs specific, measurable benchmarks.
All right, let's get into it. We're stepping into the science behind keeping us safe every day through heat.
Okay, so maybe the most fundamental place to start is how microbes die when you heat them. It's not linear.
Not a steady decline.
No. When you hit a population of microbes with lethal heat, they actually die off exponentially.
Exponentially. So, like, the death rate depends on how many are still alive.
Yeah.
It slows down as the numbers drop.
Exactly. It follows what's called first-order kinetics. It's a bit like certain chemical reactions work.
Okay.
In any given time period, a constant proportion or percentage of the remaining living cells gets killed off. It's not a fixed number dying each minute. It's a fixed fraction.
I think I see. So if you kill, say, 90% in the first minute.
Then in the next minute, you kill 90% of the much smaller number that survived that first minute, and so on.
Right. Okay.
Scientists quantify this using the specific death rate, often shown as KD. It basically tells you the fraction killed per unit time.
So a high KD means they die fast. Low KD means they're tougher.
Precisely. Low KD means higher resistance.
And if you were plotting this, say, the logarithm of survivors against time, you'd get a straight line.
You would. That straight line on a semi-log graph visually confirms that exponential decay. The math behind it shows this perfect drop-off, theoretically.
Okay, that's the foundation. But how do you take that rate, that KDD value, and turn it into something practical? Like, how long do you need to pasteurize milk in a factory?
Yeah, that's the key question. Because, early on, people tried simpler ideas. Things like thermal death time, TDT, the time to kill all the cells.
Seems logical.
Or thermal death point, TDP, the temperature needed to kill all cells in a fixed time, maybe 10 minutes. But the sources point out a big problem.
Which was?
They depended hugely on how many microbes you started with. If you start with more, it just takes longer to kill all of them, even if their basic resistance is the same. So TDT and TDP weren't really universal measures of inherent heat resistance.
I gotcha. So we needed something independent of the starting population.
Exactly. And that brings us to the decimal reduction time, the DRT, or as most people call it, the D value. This is absolutely fundamental.
The D value. Okay, what's the definition?
The DRT, or D value, is the time needed at a specific constant temperature to kill 90% of the viable cells in a population.
Killing 90%.
Yeah.
That's the same as reducing the population by 1 power of 10, right? 1 log 10 cycle.
Precisely. If you start with 1,000,000, that's 10 to the 6. One D-value time brings you down to 100,000 or 10 to the 5. Another D-value time gets you to 10,000, 10 to the 4, and so on.
Each step is a 90% reduction.
Yep. Each step is 1 log cycle down.
And crucially, because it's always 90% of whatever's left, that time interval is the same whether you started with 1,000,000 or 1,000,000,000, assuming the temperature stays constant.
That's the real power of the D-value. It gives you a measure of the organism's heat resistance at that temperature that isn't skewed by the initial load. That makes it incredibly valuable for comparing resistance and designing processes.
So graphically, the D-value relates to the slope of that log survival curve we talked about.
Directly. A shorter D-value means a steeper slope, faster killing. A longer D-value, a shallower slope, more resistance.
And the source connects this back to that specific death rate, KAD.
Right. So they're describing the same underlying phenomenon, just scaled differently. KD equals 2.303 divided by DRT. The D value essentially translates that exponential rate into a practical time needed for a standard 90% kill.
Makes it more intuitive.
Definitely. The source gives an example. If a microbe's KD at a certain temperature is 0.4605 per minute,
Okay.
then its D value, the time for a 90% kill, works out to be 5 minutes at that specific temperature. Puts a concrete time on its resistance.
That clicks. Okay, so we have this core idea. Exponential death, measured by the D value at a set temperature. But you mentioned things aren't always that simple. The source says other factors can mess with this.
Oh, absolutely. A microbe's heat resistance isn't just fixed. The conditions it's in, its own biological state, even how it's physically grouped. It all matters.
Like what kind of things? Give me some examples.
Well, we already touched on initial concentration. It doesn't change the inherent D value, but
But if you start with way more bugs, it just takes more D value periods to get down to a safe level.
Yeah.
Longer total time.
Exactly. Then there's clumping. If cells are stuck together in a clump, the ones inside are sort of shielded from the heat.
Like a little protective huddle.
Kind of. And that can make the whole population appear more resistant on your graph. You might see the death curve tail off, deviating from that nice straight line.
So the math assumes individual cells, but reality have clumps.
Reality often does. Also, the age of the culture matters. Older cells, in what's called the stationary phase, when they're not actively growing and dominating, tend to be tougher, more heat-resistant than young, rapidly growing cells.
And what about the liquid or food they're in? That must make a difference.
Huge difference. Medium composition is critical. Some things act like protectors. Starches, sugars, proteins like in milk, even moderate salt levels, maybe 2–4%, can make cells harder to kill with heat.
So sugar syrup would be protective.
It can be, yeah. Generally, any organic material can interfere a bit, and pH is a massive factor. Most bacteria are toughest around neutral pH, say 6 to 8. If you make it more acidic, lower the pH.
They become much easier to kill.
Much more sensitive, yes. That's key for food preservation.
Right. You mentioned canning acidic versus non-acid foods. That makes perfect sense now. Tomatoes versus beans.
Exactly. And perhaps one of the biggest factors mentioned is moisture. Moist heat, like steam.
Like in an autoclave.
Precisely. It's way, way more effective at killing than dry heat, like in an oven, even at the same temperature. Water transfers heat much better and helps break down cell structures.
Okay, what about after the heating? Can conditions after change the results?
That's more about how we measure survival. If some cells are damaged but not quite dead, giving them a really rich recovery medium afterwards might allow more of them to repair themselves and grow.
So it looks like the heat was less effective than if you used a tougher recovery medium.
Exactly. It's an apparent effect on the measurement. And of course, the biggest variable of all is the type of microorganism itself.
They must vary wildly.
Immensely. But the source really emphasizes bacterial endospores as the main challenge, especially for sterilization. They are incredibly resistant, dormant forms made by some bacteria. Think Clostridium and Bacillus species. They're the gold standard for heat resistance.
Which brings us back to temperature itself. You said it has a huge effect.
A massive exponential effect. Small increases in temperature can dramatically shorten the time needed to kill microbes. And just like the D value quantifies resistance at one temperature.
There's something for how resistance changes with temperature.
Yes, exactly. That's the Z value. It quantifies how sensitive a microbe's D value is to temperature changes.
Okay, the Z value, how's it defined?
The Z value is the change in temperature, usually in Celsius or Fahrenheit, needed to cause a 10-fold change in the D value.
So if you increase the temperature by one Z value, the D value becomes 10 times shorter. You kill them 10 times faster.
Correct. Or if you decrease the temperature by one Z value, the D value gets 10 times longer. It tells you how many degrees you need to change the temperature to move the D value up or down by one log cycle.
So a microbe with a low Z value, its D value changes really dramatically with temperature. It's very sensitive to temperature shifts.
That's it. A high Z value means it's more resistant to temperature changes. Its D value doesn't plummet quite as fast when you crank up the heat. The source points out Clostridium botulinum stores. Again, their Z value is pretty high, like 14 to 17 degrees Celsius. That's part of why they're such a benchmark for canning.
Let's try that example from the source material. Bacterium A has a Z of 10 degrees C. Bacterium B has a Z of 5 degrees C. They both have a D value of 20 minutes at 100 degrees C. What happens at 120 degrees C?
Okay, good example. So we're increasing the temperature by 20 degrees Celsius for both.
Right.
For bacterium A with Z 10 degrees C, that 20-degree increase is 2 Z values. 20 divided by 10 is 2. Each z-value means the d-value drops tenfold. So 2 z-values mean it drops 10 times 10.
100-fold.
Exactly. So its d-value at 120 degrees C will be the original 20 minutes divided by 100.
Which is zero point 0.2 minutes, 12 seconds.
Yep.
Down from 20 minutes.
Big drop. Now, bacterium B, its z-value is only 5 degrees C. That same 20-degree temperature increase is now 4 z-values. 20 divided by 5 is 4.
Okay, so 4 10-fold reductions.
Meaning its d-value decreases by 10 to the power of 4, which is 10,000-fold.
Whoa. So 20 minutes divided by 10,000.
Is 0.0002 minutes.
That's 0.12 seconds. Practically instantaneous killing at that temperature.
It really highlights the impact of the z-value. Bacterium B with the lower z became vastly more sensitive to that temperature jump. Its death rate just skyrockets compared to A.
So knowing the d-value at one temp in the z-value lets you predict the d-value at other temps.
That's the key application. It allows you to design processes that might fluctuate slightly in temperature but still achieve the required kill. The source also briefly mentions the Arrhenius equation, which is a more complex model relating death rate to temperature and activation energy, often used over wider temperature ranges.
Okay, this is great. We've got the exponential death, the d-value for resistance at one temp, the z-value for change in resistance with temp, and all the complicating factors. How does this translate into practice? Pasteurization, canning, medical sterilization.
Right, let's connect it to the real world. Pasteurization first.
Which, as you said, isn't total sterilization.
Correct. The main goal of pasteurization is to kill specific harmful pathogens, the ones most likely to cause disease from that product, and also just to reduce the overall number of spoilage microbes, the bioburden.
And ideally, without wrecking the product itself.
Exactly. Minimum damage to taste, texture, nutrients. Milk is the prime example, of course. The targets there, according to the source, include things like Mycobacterium tuberculosis, Coxiella burnetii, which causes Q fever, and Brucella.
And the methods?
The old way was 63 degrees C for 30 minutes, but now HTST, high temperature short time, is common, 72 degrees C for just 15 seconds.
Much faster, less impact on flavor.
Generally, yes. It achieves the necessary safety kill, but is considered better for quality.
Okay, then canning. That feels much more intense, aiming for shelf stability.
Definitely. Canning aims for commercial sterility, meaning shelf stable. And the big bad guy, especially in low-acid foods, is Clostridium botulinum endospores.
Because of botulism toxin.
Exactly. If those spores survive and the conditions inside the site can allow them to grow, they produce that incredibly dangerous neurotoxin. Other spoilage organisms are concerns too, like bacillus spores that can survive and cause spoilage, sometimes without gas, just making the food sour.
And here's where that pH difference is make or break for canning.
Absolutely fundamental. Acid foods, think tomatoes, fruits, pickles, generally have a pH below about 4.5 or 5.
Okay.
In that acidic environment, C. botulinum spores generally won't germinate and produce toxin, and heat kills microbes more effectively anyway. So you can often process these safely using just boiling water temperatures.
But non-acid foods, like beans, corn, meat.
Their pH is higher, closer to neutral. In that zone, C. botulinum can grow if spores survive. So these foods must be heated under pressure to get temperatures well above boiling, usually 121 degrees C or higher, to reliably destroy those tough spores.
So the pH dictates whether you need a simple water bath canner or a serious pressure canner.
That's exactly it. Direct application of the science. Now, how do industries define sterile or safe in terms of numbers?
Yeah, you said sterile isn't always absolute zero microbes.
Right. In practice, especially when we're dealing with heat-killing kinetics, it's about achieving an acceptably high probability that no viable microbes survive. It's probabilistic.
How do they measure that kill?
A key term is the inactivation factor, or IF. It's simply the ratio of the number of microbes you started with, the initial bioburden, XSO, divided by the number you have left after the process. IF equals X0, XT.
So a bigger IF means a bigger kill.
Yes, a bigger reduction. Now, in the food industry, for those low-acid canned foods...
The one's risky for botulism.
Yes. The common standard is often based on achieving what's called a 12D process for Clostridium botulinum spores.
12D. Does the D stand for the D value?
It does. It means the process needs to be harsh enough to achieve a 12 log 10 cycle reduction in C. botulinum spores.
12 logs. That's reducing the population by a factor of 1 trillion.
10 to the 12?
That's the scale, yes. The goal is to make the probability of even one spore surviving in a can incredibly remote notionally, 1 in 10, 12.
And how does that relate to time? Well, if you know the D value for C. botulinum spores at your processing temperature, say at 121 degrees C, the source gives it as about 0.21 minutes.
Okay, 0.21 minutes for a 90% kill at 121 degrees C.
Then to get 12 logs of kill, you need to apply that lethal temperature for 12 times the d-value. So 12 times 0.21 minutes.
Which is 2.52 minutes.
Right. That minimum process time at 121 degrees C, often called the F0 value, is based directly on achieving that 12D reduction for botulinum spores.
And if your starting number of spores was higher, or your target probability of survival needed to be even lower,
then you'd need more log reductions, more Ds. The source has another example. Start with 109 spores, want a final probability of 10 to 6 survivors.
So going from 9 down to -6, that's a 15 log reduction needed.
Exactly. If the d-value was, say, 9 seconds, you'd need 15 times 9 seconds or 135 seconds of processing time. It always scales with the number of log reductions required.
Okay. And the medical world. They must have similar standards for sterilizing instruments.
They do, often even more stringent in terms of probability. They talk about the Sterility Assurance Level, or SAL.
SAL. What does that represent?
It's the probability of a single viable microorganism surviving on an item after sterilization. The widely accepted standard, like the European EN 5556 standard mentioned, demands an SAL of 10 to 6.
1 in a million chance of a single bug surviving on a medical device.
That's the target. And again, it's often based on killing highly resistant bacterial endospores. For steam sterilization, Bacillus stereothermophilus is a common test organism because it's very heat resistant.
And its d-value like?
At 121 degrees C, the source suggests a typical d-value is around 15 seconds.
Okay, so let's run the numbers. Say you have a device with 100 spores of B. stereothermophilus on it initially. D-value is 15 seconds at 121 degrees C. How long to sterilize to get that SAL of 10 to 6?
All right, starting bioburden is 100, which is 102.
Right.
The target SAL is 10 to 6. To get from potentially 102 survivors down to a 10 to 6 probability of even one survivor, you need to reduce the population by a factor equivalent to 10 to. That is
2 minus -6, that's 10 to the power of 8.
An 8-log cycle reduction is needed. Since each d-value gives you 1 log reduction, and the d-value is 15 seconds,
You need 8 times 15 seconds.
which is 120 seconds, or 2 minutes, at 121 degrees C. That's how the required time is calculated based on the initial load, the organism's resistance, d-value, and the target SAL.
The math is clear. But the source raises an interesting point about whether B. stereothermophilus is always the most relevant challenge organism in a real clinical setting.
That's a really important practical consideration. B. stereothermophilus is used because it's tough and well studied for heat, but it actually grows at very high temperatures, so it might not be the typical contaminant found on medical devices before sterilization. The actual bugs might be less heat resistant, but perhaps more relevant clinically if they did survive.
And it mentions something called the minimum infectivity number, MIN.
Yeah, the MIN. It's an estimate of how many viable virulent microbes of a specific type you typically need to encounter by a specific route, like ingestion or injection, to have a 50% chance of actually getting infected.
And this number varies a lot.
Hugely. For something like Salmonella typhi causing typhoid fever, the MIN might be relatively low, maybe 1,000 to 10,000 cells ingested. But for, say, Pseudomonas aeruginosa causing an eye infection if splashed, it might take a couple of thousand cells for a healthy person.
So the implication is, even if sterilization wasn't perfect and a tiny number of microbes survived below the MIN, they might not actually pose a risk of infection.
That's the idea the source floats. A few surviving organisms don't automatically equal disease. It depends on the organism, the patient's immunity, the site.
Yeah.
Lots of factors.
What? There are caveats.
Big ones. The source highlights modern challenges like complex medical devices scopes with long, narrow channels, for example. These are hard to clean and sterilize thoroughly. And then there's the issue of biofilms.
Those slimy layers microbes form.
Exactly. Microbes living in a biofilm are encased in this protective matrix, and they can be dramatically more resistant to heat, chemicals, everything, compared to free-floating individual cells.
So even if this SEL calculation works for individual spores, it might not be enough for a hidden biofilm.
That's a major concern, yes. And it's driving research into other methods like electron beam irradiation or microwave sterilization, especially for complex devices where heat or liquid chemicals might not penetrate effectively or could damage the device.
It really shows that hitting those stringent SLL numbers isn't just about the math, it's about dealing with real-world complexities.
Absolutely. It's a constant interplay between the predictable kinetics we've discussed and the messy reality of microbes and materials.
Okay, so wrapping up this deep dive, it's clear that killing microbes with heat isn't guesswork. It's a science governed by predictable exponential kinetics.
And we've seen how essential those measures like the D value and Z value are. They let us quantify resistance and understand how time and temperature interact.
And then how those scientific principles become practical safety standards, like the 12D concept for C. botulinum in food, or that demanding SIL of 10C6 in the medical field.
It really puts into perspective the precise science and, frankly, the effort involved in making sure the food we eat and the medical care we receive are safe from microbial threats.
It's a complex system, but as you said, kind of elegant in how it builds on these core principles of microbiology and thermokinetics.
Definitely.
So here's a final thought for you, our listener, to maybe chew on, especially thinking about that minimum infectivity number and the biofilm challenge. Given that achieving an SL of 10 to 6 requires pretty intense processing, are we potentially over-sterilizing some things, considering many pathogens need a decent number to actually infect someone? Or are those hidden risks the resistant biofilms, maybe rare but highly virulent bugs, we haven't perfectly accounted for significant enough that these incredibly stringent standards are absolutely necessary as a crucial safety buffer, even if the MIN for common bugs is higher?
Yeah.
It really highlights that ongoing tension, doesn't it?
Yeah.
Between the established science, the practicalities of processing, and how we manage those real-world, sometimes unpredictable risks.